Hello, my name is Roderick L. Rowe, and today I will be giving an overview on the T lymphocytes. We are talking about the acquired immunity or the third line of defense. As you can see in our flow diagram, we have the B cells and the T cells, or B lymphocytes and T lymphocytes. Where do those cells come from? They come from the bone marrow. The bone marrow has a stem cell that will differentiate and turn into either of those lymphocytes, B or T. T lymphocytes make up about 75 to 85% of all lymphocytes in the blood. We have three types of lymphocytes. We are only going to talk about the ones right now that are matured in the thymus. In the picture, I have highlighted what one would look like under the microscope. If the T cell is involved in the response, it is called cell-mediated immunity because the actual cell is going to help kill. When we go to the B cells, they will secrete antibodies. So they're now called cell mediated. So in the picture below, I have different types of of T cells. We have the memory cells. We have the cytotoxic cell. We have the suppressor cell. We have the T helper cell. The T lymphocyte and its receptor will be created in the thymus. So remember, it will be originally made in the bone marrow. Then it will be released from the bone marrow, travel in the bloodstream until it reaches the thymus. And the thymus, it will develop its receptors that we're going to talk about. So the T cell was made in the bone marrow and it is released into the bloodstream where it travels to the thymus where it will mature. In the thymus, it through transcription and translation, it is going to produce a receptor. Now there's a gene or genes that make this receptor, and there's many combinations of protein that it can make, and this protein is the T cell receptor. So each T lymphocyte is going to produce 500,000 copies of a single protein that will be located on its cell surface. And that protein is called the T cell receptor. This slide shows how it will be produced. So each of those T lymphocytes will randomly choose a combination of exons. Remember, previously we talked about RNA splicing or alternative RNA splicing. So if you look at the picture, we have 4 exons in this picture. Introns will be cut out. And then you have 4 pink exons. Those exons can rearrange. The first one, you deleted the 4, and whereas the second one, you deleted the 3. This makes 2 different variations of a T cell receptor. So each T lymphocyte has its own receptor that will bind to one thing. Here is one example. Everyone has been exposed to dog dander. You have a T lymphocyte in your bloodstream that has a receptor that binds to dog dander. Here is cat dander. Notice the receptor is a different color. So this T cell receptor will bind to cat dander. This yellow T cell receptor will bind to cedar tree pollen. And this receptor will bind to peanut. So 5 characteristics of the T cell receptor. Number 1, receptor diversity is generated by the rearrangement of gene segments, or better known as alternative RNA splicing. Every possible combination occurs, leading to a huge assortment of different T cell receptors, such as binding dog dander, binding cat dander, binding cedar tree pollen and peanut. A closer look at the structure of the receptor. It has 2 regions, a changeable region and a non-changeable region. The portion that changes is called the variable region. That's the portion that's at the top that binds to the dog dander, the cedar from the tree. Whereas the constant region is the portion that binds to the cell membrane of this T lymphocyte. The constant region is embedded within the membrane of the T lymphocyte. In the picture, you're looking at the membrane on the bottom portion, the bottom sphere that you see with all those phospholipids, and the alpha and the beta receptor is anchored into the membrane of the T lymphocyte. The portion that is anchored is the constant region. I'm pointing 2 arrows at the constant region. I'm now pointing 2 arrows at the variable region. This is where the dog dander or the peanut is going to bind. So this is the variable region. The fifth characteristic of the T lymphocyte receptor is that they are relatively small, they remain attached to the membrane, and are never secreted. We're going to talk about a B cell receptor later, and it will be secreted. Along with the T cell receptor, some T cells also have another receptor called the cluster of differentiation, or CD. So in the picture along the top, I've highlighted the T cell receptor that is on the T cytotoxic cell, and it also has a receptor right beside it called CD8. If you look to the picture to the right, the T helper cell also has a T cell receptor, but it has a different CD receptor. It's CD4. Over 300 CD molecules have been named. We will only talk about CD4 and CD8 extensively. So the most important ones are CD3, CD4, and CD8. I found a picture to help show you uh, how these CD cluster of differentiation receptors are right beside the T cell receptor. As I stated, we will only talk about CD4 and 8. CD4 is an accessory receptor that is found on the T helper lymphocytes. The T helper regulates the activity of B cells and T cytotoxic cells. In the picture, I am pointing to how the T cell has the receptor and it is bound to a macrophage. CD8 is on the T cytotoxic lymphocyte, which directly kills other cells. That is the circle that is green. So the T helper cell is going to communicate with other cells and tell them what to do, whereas the T cytotoxic cell is going to specifically kill other cells. T lymphocytes contain two receptors. T receptor, T cell receptor, and the cluster of differentiation. This is an overview of the production of those T lymphocytes. They started in the bone marrow. They're going to mature in the thymus and then eventually start to circulate throughout the body. And they're going to be normally housed in those secondary lymphoid tissue.